I'm in now.
Okay. This is
Hi, Banu.
Hi, Bjorn. How are you?
Fine. How are you?
Good. I am
Looking forward to your presentation.
Well, if I can pull this out, I'm still waiting for this to update. If I have a problem, I'll ask you to go first.
Okay.
Hello, Bjorn.
Hi. Hi.
Hi. Hello, Dr. Banu, Dr.
Good
Bjorn.
to see you again.
Good evening. Good morning. And good evening, Dr. Banu.
It's 1
Hi,
p.m. So it's 1 p.m.
Hello. I'm audible?
Yeah, you're audible.
Yeah, we can hear you.
I could not hear anything.
So Naresh, discuss with our director sir, and then we can start.
So
link is same, Manoj, or today there is new link?
Same link.
Same link, okay.
So Naresh,
Hello. Now I'm audible?
hi.
Yeah, you're audible.
Yeah, audible.
Okay. Hello. Achha. हेलो हाँ सर, वो एक इवेंट था ना आज भी जो सेकंड डे है, उसको कर दे सर। नहीं, अपने रिमार्क वगैरह अब, क्योंकि आप कोई नहीं है सर, एक बार शुरू कर देगा फिर वो 5 मिनट के लिए थोड़ा सर कर दीजिए। सेकंड डे ना, नए इसमें बियॉन पीटरसन है सर जर्मनी से, और भानु तेलुगु है यूएसए से। हाँ अच्छा ओके उसको शुरू कर दे, या धीर सिंह सर को बोल दूं मैं एक बार? धीर सिंह से अच्छा ठीक है सर।
So Naresh.
Hello.
Can you hear me? Hi, Naresh.
Yeah.
My presentation is not updated on my iCloud yet, so we may ask Bjorn to go first before me.
Yeah, Bjorn will go first before to you, and if
there
is difficulty, so you can share the presentation to me, and then I can upload from here if there is any difficulty in uploading.
Okay, that sounds good. Should not be a problem. All right, that will work out. Bjorn, go first.
Yeah, we will start because director sir has been busy in some other meeting assignment. So just I was waiting for the joint director, Dr. Dhir Singh. If he's been allowed, he will give us the signal to start. We will start immediately. Okay.
That sounds good. This is weird.
Banu, for you, it must be quite early in the morning, huh?
Yeah, yes, it's 6 o'clock in the morning. Yeah. Yeah, technology. So I put it on the iCloud drive and it's not updated yet. So
the cloud is still sleeping. It's too early in the morning.
Yeah, I guess so. Technology can be a problem sometimes.
Yeah, technology is for the problems to solve that we would not have without using it.
How are things with you guys there?
Oh, quite good.
Good. Good, good.
Yeah.
Is
We have
Heiner
now
still
some-
at your institute? Is Heiner still
Oh
at your institute?
no, he retired
Oh, he did.
3 years
Okay.
ago. 3 years
Hello.
ago already.
Hi. Hello, I'm audible?
Yes, Naresh. you are
Yes. Okay, very warm good evening to Indian participants and good morning to Dr. Bhanu and from the participant from USA, and good afternoon to Bjorn and his participant from the Germany and from Europe. It's indeed a great honor for us. But today we have the very eminent scientists who have been working on particularly on the assisted reproduction and the genome editing in the farm animals, and they have these extensively used these genome editing tools, particularly CRISPR, to produce the model animals in pigs as well as in the cattle. So since today the Honorable Director NDRI, Dr. M.S. Chauhan, and the joint director, they have the meetings, so they told you continue, and they may be in between, they may join. So before this starts, now I invite the head of the department, Dr. Sachinanda Dey, for his opening remarks, and then we will have the presentation from the Bjorn Peterson.
Naresh, you can introduce, then
Okay.
can read this his contribution on this, both Bhanu and me.
Okay. So the first presentation we have the Bjorn Peterson. He studied the veterinary medicine at the University of Veterinary Medical Medicine Hanover in Germany from 1997 to 2002. Currently, the designation is the Doctor Med-Vet, and his department is the biotechnology. After receiving the probation, he started his doctoral study at the Institute of Farm Animal Health. During his study, he established a pig cloning protocol and started performing targeted gene modification of porcine genome by somatic cell nuclear transfer and homologous recombination. He received his doctoral degree in 2004, and since then he worked as a member of the permanent staff at the Institute of Farm Animal Genetics, FLI Institute, Marenzi. His main research topic is the use of the DNA endonucleases such as zinc finger nucleases, CALN, and CRISPR to alter the genome of the livestock species in order to address the future challenges in animal breeding. His current projects are focusing on the xenotransplantation, foal cattle, genome sexting by CRISPR-Cas. And as currently he has a 151 publication and total citation index at the Scopus Index 26. And is a major scientific achievement is the first biallelic knocking out of the endogenous pig gene using the zinc finger nucleases. And the technology, as I mentioned, he has been worked on the somatic cell nuclear transfer in pigs, genome editing by homologous recombination, and ZNF and the CRISPR-Cas. So now I invite the head of the department, Dr. Sachinanda Dey, for his opening remarks, and then to the Bjorn Peterson for his talk.
Okay, I think good afternoon to all of you. So today we are having 3 lectures on this CRISPR-Cas. Yesterday we have listened about poultry and this sheep and goat, and today another important animal in the pig sector. I think he is going to give about not only CRISPR but zinc finger, which is started this genome editing in this particular aspect. And another taste of this thing that animal science is going to reach the human health in terms of genome transplantation. That is also another area, I think. And then we'll be having lecture of Bhanu Telugu. He will be talking about genome editing in case of cattle. Then Naresh will little bit discuss about whatever work we have started in our Animal Biotechnology Center, NDRI. Well then, without taking any time, I am requesting Dr. Peterson to please you share your slide and start this lecture so that we can able to make this today's presentation in time. Okay, over to Peterson, please.
So you can see my presentation now in full screen?
Yes, it's visible.
Yeah, great. So thank you very much for this nice introduction. It lets remind me how old I have become so far. So it's good to see some familiar faces like Naresh. And it's a great pleasure to give a talk about the projects that are currently going on here at the Friedrich Loeffler Institute in Germany. So the title of the talk is Applications of Gene Editing in Pigs at the FLI. And I would like to give you a short introduction and then introduce our current projects here at the FLI to you. So as you all know from the talks yesterday, and for all these guys and ladies working on genome editing, we have now these CRISPR-Cas tools in our hands with which we can modify the genome of our livestock species quite easily by just introducing a double-stranded break in the DNA, which is then repaired by the non-homologous end joining pathway for introducing indels in the genome, which leads to a functional knockout, or we can use two CRISPR molecules to induce some large deletions, which can be used for a functional knockout, or can be used to excise some sequences from the genome, or you rely on the homology-directed repair pathway to introduce specific sequences that you provide with a template that are usually then used for a gain of function in these livestock species. So we have three different ways of modifying the genome of our livestock species in our hands. So one is that you use zygotes and micro-inject the CRISPR-Cas plasmids or RNPs, for example, into the zygote, and then transfer these micro-injected embryos back to synchronized recipients. And your technology is the electroporation of zygotes, where you introduce the CRISPR-Cas molecules into a zygote by electroporation, and then transfer these embryos back to synchronized recipients. Or the most used way so far is the somatic cell nuclear transfer, where you transfect the cells, do the genetic modifications on the cells, and then use these cells as donor cells for somatic cell nuclear transfer to produce cloned embryos with the desired genetic modification and transplant these embryos back into your recipients. And then you get the genetic modified offspring from this approach. So usually we rely still on the somatic cell nuclear transfer because it has some advantages over the other technologies. But in some cases, for example, if you just want to introduce a knockout of a gene, the microinjection pathway is also a very common way to use. So what are the main applications of genome editing in livestock species? So one is for agriculture. So here are some projects or some publications regarding poultness, especially in cattle, of course, not in pigs, to increase the growth or the development of the animals. You can also adjust the amount of lactation or the ingredients in milk producing animals. The wool or meat production can be increased by genetic modifications. That was presented yesterday by Xiaolang Wang, for example. Or you can use it for reproduction, genome sexting. This is something that I would like to show you. And also for environment purposes, to make pigs, for example, more environmentally friendly, or for dietetic improvements of the meat, for example. A huge application for genome editing in the pig especially is biomedicine, but you can also apply to milk producing animals for gene farming. That means that you produce pharmaceutical active substances or molecules in the mammary gland of these species. You can use it for human blood substitution or xenotransplantation, which I would like to introduce to you. And a huge application is to provide human disease models, especially in the pig. But we use it also for basic research to analyze epigenetic reprogramming, for immunological questions, or to find ways to make livestock more disease resistant, for example, against African swine fever or PRRS, which is the economically most important disease in pig production. So I would like to introduce you the Sexting project that we have by genome editing, xenotransplantation, disease resistance, and an immunological project that we have run in the past. So first, sexting by genome editing. So in most of our livestock species, one gender is preferred because the desired product is produced by only one sex. For example, milk is produced by females and eggs also from females. Males are mostly used for meat production, or you have a higher gain for the product for one sex. And sometimes males are not the desired sex because threat to farmers and conspecifics. So that is also the case why maybe females are preferred in some of these species. In the pig especially, and about 75% of consumers are sensitive to boar taint. Especially women and certain ethnic groups show higher degree of sensitivity. And this comes from the breakdown products of tryptophan, which is scatol, which exhibits an intense fecal odor, and androstenone, which is mostly related to a urine attribute. So this is shown here. So scatol is a tryptophan metabolite. It is synthesized in the colon by microbial degradation, and it's absorbed from the large intestine and circulates in the blood, and it's stored in adipose tissue. Hepatic degradation is inhibited by androstenone and testosterone. And androstenone, for example, is produced in the testes. It is a testicular steroid with urine smell, and it's produced in Leydig cells regulated by the hypothalamic pituitary gonadal axis, and it can be catabolized by the liver, stored in adipose tissue, or excreted in saliva. So to avoid boar taint, usually our piglets, the male piglets, are castrated without anesthesia at a very young age, up to day 7 after birth. And this is routinely practiced in many parts of the world. About 75% of male pigs in the EU are castrated without anesthesia. But this causes also some animal welfare issue because it causes pain and stress and also risk of infections. So, and therefore castration without anesthesia is prohibited in Germany since this year, January this year. So especially the German farmers had to find some alternatives to the castration without anesthesia. And one alternative is the castration with anesthesia. Isoflurane is the
Hello. Beyond, your slide has been unshared. So there is no. Okay.
Yeah.
Okay. Maybe some internet connectivity issue.
Also on mute.
Beyond, you are muted. Unmute yourself. Okay.
So now I'm on again. Yeah.
Yes.
So there's some cons with the castration with anesthesia as well. The additional costs, the greenhouse, isoflurane is a greenhouse gas. The safety margin is very limited. So, and the workers report headache and dizziness when they use it. So another alternative would be boar fattening. It has a lot of pros, like no pain for the piglet, reduced feeding costs, higher muscle content, and increase in unsaturated fat. But there are also some cons, like restless and dominant pigs, penile injuries, and detection of boar taint is necessary at the slaughterhouses, which is very cost intensive. So another option is the immunocastration. It is like a vaccination against gene RH. It decreases the aggressive mounting behavior of the boars. They behave like castrates, and the overall growth rate is higher. But it had also some cons, like additional costs again. There's some low responder to the injections, so that still produce androstenone and scatol and have some boar taint. 2 shots are necessary, and there's also some risk of self-injection of the farmers. So we were thinking maybe of another alternative in that perspective. How could genome editing maybe help, or find some other ways to do it? We found one target, which is the sex determination by the SOY expression from the Y chromosome. And there's some mouse data that suggests some certain role of SOY in the testis formation. So when it's expressed at a special time point during development, it serves as a transcription factor and increases the expression of SOX9 and FGF9, which then leads to testis formation and to a male phenotype. So without SOY expression, other genes are upregulated, like WINT4 or SPO1, and that leads to an ovary formation or ovarian development, which results in a female phenotype. So the time of SOY gene expression is quite limited. So in the mouse, it is from day 10 to 12.5. In the pig, there's not so much data about it, but the suggestion is that it is expressed from day 21 on, and the highest expression rate is during day 21 and day 23 after insemination. So, or of gestation. So first, we started to find the SOY and find the sequence of the porcine SOY in our pigs, and we found some quite interesting results. And that was, we used a digital PCR, and what we found is that we do not have a single SOY gene on the Y chromosome in the pigs, but we have a duplication of the SOY gene. So we compared it with a gene that is B allelic, like the GGTA1 locus, and we got the same amount of positive results from the SOY gene, while another gene that is located on the Y chromosome is only there at a single copy. So that was the first results that we obtained, that we have a duplication of the SOY gene on the Y chromosome of the pigs. And this is displayed here. And so we generated, or we tested guide RNAs for the SOY gene that excise the whole HMG box of the SOY gene, which is the functional box of the SOY gene, and it should excise it and lead to a deletion of about 300 base pairs in the SOY gene. So the first thing that we did was using an RNP CRISPR vector and with the 2 guides that lead to excision of the HMG box of the SOY gene, and injected it into the cytoplasm of in vitro fertilized pig oocytes, and then transferred these embryos back into the recipients. So 3 recipients received these embryos, in an average 32 embryos per recipient. All of them became pregnant. 1 resorbed after day 25 of gestation. The other 2 remained pregnant until delivery, and they delivered in total 12 piglets. All of them were female. So we did not use any sexed sperm. So we were expecting that some of these offspring were males, but all of them were females. So, and then we took some samples and ran a specific PCR for the SOY gene. And in 3 of the offspring, we detected a product. So these 3 piglets. So that means that they should be males and not females. And we also did a karyotype analysis of these pigs, and we could clearly detect a Y chromosome in these pigs. But when you look at them, you can clearly see that they have a female phenotype. So they have a male genotype, but a female phenotype. And we also sequenced then the SOY locus, and we detected the deletion that we expected. In this case, it was a little bit shorter than expected, but that was the deletion that we expected of 292 base pairs. And we also did then use then cells that we transfected with our CRISPR constructs targeting the SOY gene. And we first did a PCR to detect the deleted SOY gene and use these cells for somatic cell nuclear transfer to produce some more offspring. And 2 of the recipients became pregnant and delivered in total 6 piglets that were all females. And we used a male cell line, so that was also the final proof. And all of them again displayed a female phenotype, as you can see here. And we also analyzed the internal genitalia, first at day 34, and they also developed internal female genitalia, like in uterus and also ovaries. And when you compare it to a wild type pig at that age, there's only a slight difference in the size of the ovaries. The SOY knockout pigs had smaller ovaries at that time point compared to wild type pigs. But this difference became much more obvious when we compared these animals at an age of 90 days. So at 9 months, 9 months. So you can see they have a much smaller vagina compared to a wild type. So the uterus is quite immature compared to a wild type control at the age of 9 months. And also the ovaries are much smaller and they do not have any functional follicles on the surface of the ovaries. So we also did a immunological or histology analysis of the ovaries. So this is at day 34 again. So they did not differ to the wild type controls, but again, at the age of 9 months, we could not detect any follicles in the ovaries compared to a wild type control, where you have here the follicles that are built in the ovaries. So these pigs show no further development and maturation of the inner and outer genitalia and no follicles in their ovaries, which results in infertility. So we try to inseminate these pigs. We stimulate them with hormones, but they do not respond to this hormonal treatment and they became not pregnant. So these pigs are infertile. But nevertheless, we also wanted to know if they have some maybe some sort of follicles or oocyte development occurring in the ovaries, and that was shown here. So we found some FOXL2 positive cells in the ovaries of these SOY knockout pigs, and FOXL2 is an ovarian marker which can be detected in the ovaries of these SOY knockout pigs. So at least I would say they have the potential to produce matured oocytes, but at this time point, we do not know what is necessary to fully mature the ovaries and the uterus. Nevertheless, all these results that I presented you here were published this year in PNAS, and we have now some more plans with these pigs. So one question, or what we want to try to find out, is to elucidate the miracle of sex development. So which other factors are necessary for the maturation of the uterus and ovaries? So what other gene expressions, for example? So we would also like to produce X0 animals to investigate whether gene expression from the remaining Y chromosome hampered the female development. Maybe there's some disadvantages. Maybe some genes are still transcribed from the Y chromosome that hamper the female development. And does the absence of a second X chromosome influence dose-dependent expression of X chromosome-specific genes normally supporting female development? And what we also want to try, because I introduced the problem with the castration. So we would like, and we have some ideas in our head, how we can achieve this. Is the generation of a boar that produces only female offspring feasible by this approach? So this would be the final goal at the end, but at least it's a lot of basic research that we would like to do. And also vice versa, to do the whole project in the opposite, is to produce females that express SOY by introducing it as a transgene. And do they show a male phenotype then? And what is the outcome of this? Do they produce also sperm cells or not? Are there also other genes necessary to be expressed for this? So this would be quite interesting. So this was the first project that I wanted to introduce to you. The second is a more immunological project because, well, we are the Federal Research Institute for Animal Health, and we have a lot of immunologists in our institute. So that was to produce pigs that lack gamma delta T cells. So gamma delta T cells are still a miracle because the function of the gamma delta T cells and their contribution to protection against infections is still unknown. There are higher numbers of circulating gamma delta T cells compared to mice in the pigs, which render it a suitable animal model. For example, in mice and humans, you only have 0.5 up to 10% gamma delta T cells circulating in the blood. And in the pig, you have about 20 to 50%. So the question is, why is that the case? What kind of role do they play? So therefore, the availability of a gamma delta T cell deficient animal model may add important new insights in the role of this part of the adaptive immune system. So for gamma delta T cells, we have identified a conservative region of the TRDC gene, and we targeted the exon 4 of that gene and injected, again, a CRISPR-Cas RNPs that target the TRDC locus. So again, we introduced a deletion of 40 base pairs. So we transferred these embryos into 2 recipients. Both of them became pregnant. And in total, 14 piglets were born. 6 of them were completely, or showed a complete TRDC knockout, which is displayed here. The good thing is when you introduce deletions, you can easily detect it by a simple PCR. And we further sequenced the TRDC locus in these animals, and we could clearly show that they have a very clear 40 base pair deletion at the TRDC locus in the exon 4. And for further experiments, we wanted to produce some syngenic controls to the TRDC knockout pigs. Therefore, we used the cloning approach, and we transfected cells with a plasmid containing the Cas9 and the 2 guide RNAs, and then selected these cells for a complete knockout of the TRDC. And this is a mixed population. So our assumption was if we use this cell clone for cloning, we might get some pigs that still have gamma delta T cells and some pigs that have a B allelic knockout. And indeed, this was the case. So we got several pigs that had a 40 base pair deletion at the TRDC locus. This one had a little bit larger deletion of 45 base pairs. And we had some piglets in the same litter that had no modification of the TRDC locus. So this is very important if you perform some immunological studies, which also rely on the MHC class 1 phenotype of these animals, because that has a huge impact of the outcome of these experiments. So in this case, we are now in the lucky position to have modified pigs that have the same genetic background than the wild type control group. And we also did some FACS analysis of blood cells from these animals. We could clearly show that they do not have any gamma delta T cells anymore. And all the other compartments of the immune system, they were not affected by this gamma delta T cell knockout. So we are now starting some new experiments in the future. How do they react, for example, to a vaccination? Do they produce the same amount and titers of antibodies and so on, to find out what the specific role of these gamma delta T cells is. And yeah, these results that I showed you today, they are going to be published on Thursday in scientific reports. So if you want to get some more information about these pigs, you can find this in this article published in 2 days. So as I mentioned, we are some viral institute. So we are also working on disease-resistant pigs, and you have several options to protect pigs, for example, from viral diseases. Important roles play receptors for the viruses to enter the cells and infect the cells. Otherwise, you can also use genes, transgenes, to make the pigs more resilient or resistant against some viruses, or you can modulate the immune system to make them stronger to handle with the virus. So for that purpose, we performed a lot of studies with in vitro host pathogen interaction studies, which leads to a hypothesis what kind of strategy we should use for protecting the pigs from some specific diseases. And also the other things were done to find maybe some good hint where we could use gene editing to make them more resistant. And one of the most important diseases at the moment for our pork industry here in the EU is African swine fever. So African swine fever is endemic in Africa, as the name says. It's endemic in warthogs and bush pigs. They only show subclinical signs of this infection. They are natural hosts, and it's transmitted directly or via arthropod vectors. In domestic pigs, they are fully vulnerable to this disease. They develop an acute, rapidly fatal hemorrhagic fever. It's some sort of a pro-inflammatory cytokine storm driven by infected macrophages. And you can see a widespread apoptosis of infected macrophages and uninfected lymphocytes. And usually these pigs are 100% susceptible to this virus, and you have a mortality rate of more than 90%. So it's a very, very dangerous disease for domestic pigs. And the problem is that we have a huge outbreak of African swine fever here in Europe, but also in China. So there are some estimated numbers for China we have no official numbers, but it is expected that they killed about 150 million pigs during the last 2 years due to the African swine fever outbreak. So the problem with that disease is that there is no vaccine available. It's a very huge virus. It has very high mortality. So the only option to prevent this disease from spreading is culling of infected pigs. And since September 2020, we also have the first cases here in Germany, and only 3 days ago, we had also the first outbreak in domestic pigs here in Germany. So the question is, what can we do? And it came into my mind that CRISPR protects bacteria from being infected. And I was thinking about, and others did that as well in plants, for example, can't we use that system for making organisms resistant against viral diseases? So we constructed a transgenic vector that encodes for Cas9 and for guide RNAs targeting the African swine fever virus, which belongs to the Asfaviridae family and is a double-stranded DNA virus. So the perfect target for CRISPR-Cas. We produced, at the beginning, we only produced some transgenic cells and did some in vitro studies, and then used cells to produce transgenic offspring. So these are the results from the in vitro infection studies. So we targeted a very conserved gene, which is necessary for replication of the virus when it has infected the cells. So we transfected wild boar lung cells with our construct and then infected them with African swine fever virus, different strains. So the BA71 strain is a laboratory strain, and the guide RNAs were designed to target the P30 gene of that particular strain. And what you can see here, so the red dots are infected cells. You can clearly see that these cells are much better protected against infection of this virus compared to wild type lung cells. But what you can also see is that they are still susceptible to the Kenya strain. And these cells were also protected against the Armenia strain, but again, susceptible to the Kenya strain. And then we looked into the P30 gene of the Kenya strain, and we found 4 mismatches in that area. So which made it quite obvious that this virus is maybe resistant, or the guide RNAs could not bind to that specific sequence. So we observed almost complete protection against infection with the laboratory strain and Armenia. And we saw some escape mutations against Kenya 1033 because it has 4 mismatches at the targeted region, which avoids CRISPR-Cas binding and cutting. So the CRISPR-Cas expression in these cells leads to 10,000-fold reduction of viral replication, which should provide protection in vivo. So we have now started with initial in vivo experiments, which did not result in resilience or resistance of the modified pigs. So we are now trying to improve our vector regarding expression levels, tissue specificity, and also integrate some more guide RNAs, which should also make these cells resistant against infections with the Kenya strain. So these are the piglets that are currently born, which are our new great hope for these infection studies, which will take place at our headquarters of the Friedrich Loeffler Institute, located on the Isle of Reims. They have a biosafety level 3 lab for large animals where they can perform these infection studies with these very dangerous virus. They also work with Ebola viruses and so on. So they have the permission to do these infection studies. So we are quite looking forward to these experiments in the future. So the last project I want to talk about is xenotransplantation, which means we want to use organs from the pig as organ donors. And this is the case because worldwide, you have a very huge waiting list of patients that are waiting for a transplant of a suitable organ. And this is some data from Germany from the days 2010 to 2019. And you can clearly see that there's a huge gap of people that are on the waiting list waiting for a suitable organ, like here, kidneys, for example, leading to the fact that each day 3 patients in Germany die while they are on the waiting list for an organ transplant, and the average waiting time for a kidney transplant is 5 to 6 years. So there are some approaches for organ replacement, like allotransplantation, which you all are familiar with. Also tissue engineering with using embryonic stem cells or iPS. Also chimeric pigs, where you produce human organs in the pigs, which is still at a very preliminary stage, and xenotransplantation. So the pig seems to be the perfect organ donor because it can be kept under highly hygienic conditions. The genome reveals high similarity to the human genome, and also it has the physiological parameters are similar to humans. Also the organ size, there are less ethical concerns because we use it for meat production for a long time now. It has short generation intervals, rapid growth, and the transmission of pathogens is meanwhile considered to be of minimal risk. But there's a huge problem, that are severe rejection response, which originate from the fact that the pig still expresses some galactose epitopes on the cell surface synthesized by the alpha-1,3 galactosytransferase. So humans and old world monkeys don't possess gal epitopes on the cell surface anymore. We just have a pseudogene in our genome. So we lost it during evolution, which might be an advantage because also bacteria have these gal epitopes on the surface. So maybe that made it much more simpler for the immune system to detect these bacteria. So binding of preformed antibodies, about 1 to 3% of all circulating antibodies in your bloodstream right now are directed against gal epitopes. They activate the immune system and leads to the hyperacute rejection of the xenograft within minutes. But now with the genome editing tools in our hands, we can add some genetic modifications to the pig genome. For example, knocking out some xenoantigens like the galactosytransferase, which is responsible for the gal epitopes. But there are also some other candidates which produces some sugar molecules on the cell surface of pig cells, which need to be knocked out. Otherwise, you also have to overcome some incompatibilities between, for example, the coagulation system. So therefore, you also have to express some transgenes, like, for example, thrombomodulin. And you also have to protect the endothelium from becoming activated to prevent clotting in the organs. So this is some strategy which genetic modifications might have to be added in the pig genome to achieve a transplantable organ in the pig. So one of my most favorite transgenes I would like to introduce here to you, which is Hemoxygenase 1. It is an enzyme that catabolizes heme and produces some products like free carbon monoxide, free iron, and biliverdin, which all have some properties that might be beneficial for xenotransplantation. And we tested organs of a simple Hemoxygenase 1 transgenic pig in an ex vivo hemoperfusion system. That means you perfuse this kidney, this porcine kidney, with human blood, and you can measure several parameters like urine production, intravascular resistance. And this is displayed here. So this is a simple hemoxygenase transgenic kidney. It still have these gal sugars on the cell surface, and it's now perfused with human blood. So here you have this pale color because it is ischemic at the moment, and now it is perfused with human blood. You can see it becomes some reddish again. Here you have the venous backflow. Some blooding is stopped here. And you can perfuse such a kidney for the maximum perfusion time of up to 4 hours without having any other interventions, and we're still expressing the gal epitope. So this is a short video after 2 hours of perfusion. You can see that it's perfectly working. It produces urine, and it still looks very good. Looks like in the beginning. So as I mentioned, we could perfuse these kidneys for the maximum perfusion time of 4 hours. And this is how a wild type kidney would look like after about 1 hour of perfusion. So we could also show that the beneficial effects were generated by the expression of Hemoxygenase 1, because also endothelial cells were protected from becoming apoptotic after challenging them with TNF-alpha, increasing amounts of TNF-alpha, and we could reverse this protective effect by adding a specific Hemoxygenase 1 inhibitor, which is zinc protoporphyrin 9. We could completely abolish this protective effects. And yeah, this is shown here again on endothelial cells. And we also use these pig hearts in an ischemia reperfusion model, where one of the coronary arteries was included, and so there was a artificial infarct was set at the heart, and you can clearly see that the Hemoxygenase transgenic pig hearts were much better protected. And this could also be abolished by adding a Hemoxygenase 1 inhibitor. And yeah, so you can clearly see the protective effect of Hemoxygenase 1 transgenic expression. The problem is with the transgene, if you introduce some more transgenes, you have to combine them somewhere, otherwise they get segregated when you breed these animals. And we have now pigs in our consortium that is working on xenotransplantation that express 5 human transgenes and have a knockout of 3 endogenous genes. And all of the transgenes are located at a single location on chromosome 6 of the porcine genome, so they are transmitted together to the next generation, which makes it very easy to produce some more modified pigs for xenotransplantation by simple breeding. And you can here see that these pigs that have these human transgenes and have the additional knockouts are really well protected against specific lysis assay, where you add some human serum to these cells. So they are almost completely protected against any lysis from this complement system. Another strategy is to put all these transgenes at a specific locus, like the ROSA 26 locus or the GAL locus. So currently we have pigs that have Hemoxygenase 1 and CD55 integrated in the ROSA 26 locus. And the construct are designed that you can add another transgene at the same spot behind the already existing transgenes, so which is called gene stacking, so that we have maybe one day all of our transgenes that seem to be beneficial for xenotransplantation located at the ROSA 26 locus. So what we call the low carb pig is that we delete or all the sugar molecules from the pig cells that cause some xenogenic reaction, which are B4-GAL-NT2, CMAH, and GGTA1. And here are some cells that we transfect with CRISPR-Cas molecules, targeting all 3 of them. And then we selected for GAL-negative cells and use them for producing pigs. Another target here is the beta-2M, which produces MHC class 1 knockout pigs, which seems to be beneficial to protect organs from T-cell mediated rejection. So we have also produced piglets that have a beta-2M, CMAH, GGTA1 knockout. And our collaborators Immunic, they have produced a quadruple knockout pig for xenotransplantation, which carries the knockout of the GGTA1, the CMAH, the B4-GAL-NT2, and the beta-2M. So this is now the basis where we are working on to produce pig for xenotransplantation. These are some expression data of SLA class 1 in these knockout pigs. So you can clearly see that they do not express SLA1 anymore on the organs, compared to, for example, wild type pigs. And they are much better protected against human T-cell, T-cell, they are much better protected against human T-cells. So as I mentioned, we also use the microinjection technology for simple knockouts because it is fast, efficient, and it saves animals. So it's quite efficient to generate gene knockout pigs, and about 60% are biallelic knockouts, and they are much healthier than the cloned piglets. And this was one experiment where we injected these CRISPR-Cas to knock out GGTA1, and 4 of the offspring were biallelic knockouts, and 2 of the animals had some mosaic expression here, 50-50. This is more wild type than knockout, but nevertheless, it's quite efficient, and you do not have the side effects of the cloning technology. You can see these little piglets here. So if you ask me how close is xenotransplantation, so in 1995, one of the pioneers of xenotransplantation stated that it's just around the corner, but it may be a very long corner. I think the corner has become much shorter now. So these are the survival times after transplanting a pig organ into a baboon. And our consortium in Germany achieved the longest survival so far of a orthotopically transplanted pig heart into a baboon with 195 days. Orthotopic means that you replace the baboon heart with the pig heart. Now there's a preprint published by Mohamed Mohuidin, who describes survival of 264 days now. So there's some startups in the USA that want to bring xenotransplantation into the clinics. And we have also run now an application for clinical trials. So it's getting much, much closer now to the clinics, I guess. So this is the consortium, our partners in the consortium. And yes, so with that, I would like to summarize and conclude. So we have some very critical problems that humanity is facing now with the growing human population, which is also demanding better nutrition. We have the world climate changes, which affects agricultural production and the environment. Animal welfare plays a more and more important role in agriculture. And the One Health concept requires new strategies to avoid spreading of zoonosis. And as I mentioned, the demand of suitable organs for transplantation is steadily growing. So genome editing is a perfect tool that is very transformational. It's a very disruptive technology. It's easy to use, time saving and cost effective. And it can be the one of the tools that can help us solving the problems described above. So with that, I would like to thank you and to thank all my collaborators that participated in these very interesting projects. And without them, we could not achieve the results that we have achieved so far. And yes, I would like to thank you for your attention, and I'm happy to receive your questions. Thank you very much.
Thank you, Bjorn, for your exciting talk and sharing of the data from the sexting of pigs to xenotransplantation. Really exciting results you got. We have a couple of questions, so please unshare your slides so we will move to the questions. First questions from Thirumala Taluri. Do SRY knockout pig had a visible follicle on the ovary, and what were the hormonal melinine of the same? How they were inseminated? Whether they have exhibited any stress symptoms? You can also see this question in Q&A boxes.
No, they did not. So we did not see any functional follicles on the surface of the ovaries. Also, when we did the histology, we could not detect any matured follicles. We synchronized them hormonally and then inseminate them, but they did not become pregnant. So we say that these pigs are infertile.
Yeah, another question for anonymous attendee: what are the pros and cons of the genome editing practical applications in multiparous pigs as compared to the uniparous dairy cattle? These are general questions.
Yeah, I mean, the pros are that you have more offspring, of course.
Yes.
I mean, and you have a shorter gestation time, for example, in the pig. When you work in cattle, you have to wait 10 months until you get the first offspring, and you just have the single offspring. So to get some, to produce some reliable data, you have to transfer a lot of embryos to a lot of recipients in cattle. So that is much easier in pigs, and the gestation time of a pig is just 115 days. So that are, of course, the pros. I mean, the cons, I mean, it depends on what you want to do. So, for example, if you do biomedical research, the pig has a lot of pros because it's very similar regarding physiology and anatomy. When you want to do something about reproduction, then maybe the cattle is a better model because it has the same gestation time. It is uniparous like humans. So that are maybe the cons.
Yeah, there are the pro and cons of the each species. Anyway, the Ravinder Kumar from NBFGR, he's asking about does the efficiency of the CRISPR-based genome editing vary from trait to trait or species to species?
I cannot. Well, I don't think that there is such a huge difference in the efficiency from species to species. So when I compare the data that others publish in other species, I mean, they have almost the same efficiencies. You have differences in the efficiency regarding your target. I mean, we have some loci in the genome that are very easily to target. You achieve high efficiency all the time, and we have some that are really hard to target. Sometimes you have to find the right genome editing tool to get the gene edits at that specific locus. So, but we are in the lucky position now that we have so many variants of genome editing tools like base editors, several different base editors, prime editing is coming. We have different Cas constructs now with different PAM restrictions, or they are PAM free. So that that makes it now also feasible to target some hard to target genes in the genome.
Yeah, thank you. Abhishek Bharatwaj is asking, can we use this technology to create a bull to produce the female progeny, what we have done the sexing?
I hope so.
Yeah, hoping for the best.
This is something that we're trying to show in pigs. I mean, one of the drawbacks could be that our strategy would start with some sort of a transgenic bull. The offspring would not be transgenic. They would just have an edit. So, but I mean, that would be the difficulty to bring that into the breeding programs at the moment. But yeah, we're working on that. And if it works in the pig, it can be adopted to the cattle. I'm absolutely sure.
Okay, just to supplementing this, I think in your presentation, you were knocking out of the SRYZ. Any other gene you have targeted for this, how the phenotype change, or only SRY?
Not at the moment, only SRY, but we also, I mean, we targeted a lot of genes, but not for that purpose. That was used for producing X0 animals. So there we target several genes on the SRY gene to completely destroy it.
Okay, Dr. Abubakar Wakil is asking about the comparison between the zygote manipulation and the electroporation. Which one is the best result and why?
I think both work well when it's established at your laboratory. So, I mean, if you have a good expertise in doing these micro manipulation, I mean, you get really good results with doing the micro injection. Electroporation is much easier to use for many laboratories because you just need skills for getting the oocytes, mature them, and have a good IVF system maybe. And then you just do the electroporation. You do not need micromanipulators or micromanipulation skills. And especially the Japanese guys, I mean, they publish now so many papers describing the use of electroporation for zygote manipulation, and they achieved really great results. So I think it's depending on what you have established at your laboratory and what you want to do. So we are now also trying the electroporation and want to compare to our microinjection approach. And it's, I mean, with electroporation, it's much easier. You can electroporate 20 zygotes at once.
Okay, two quick responses from your side because we are running up the short of the time. So one question from Rajiv, is there any possibility to target ASF viral host receptor? Hope it is not yet standardizing instead of targeting P53 gene of ASF. African swine fever is asking about the host receptor targeting instead of the targeting the P30 gene.
So what was the question? So the question was about the receptor of the ASB, right?
Yes.
That was the first one. So there's no specific receptor. So the problem with ASB is that it uses several receptors. So, and they have also some physiological properties. So that is the problem with knocking out the receptors. So yeah, that is.
The last questions from Satish Kumar. If we inject the human embryonic stem cells into the pig blastocyst, will it produce a mixture of the pig or human organ, or it will be minimize the rejection rate of such organs in human? That is the human embryonic, what I understand, the human embryonic stem cell that is edited and injected into the pig blastocyst, it will have the human organs too. That is, yeah.
Well, the problem currently with that technique is the amount of chimerism that is achieved by this technology. I mean, there are some publications where they want to do this using currently monkey cells and so on, but the chimerism efficiency was very, very low at that time point. So maybe this could be, the reason for that could be maybe because there is such a evolutionary distance between humans and pigs. So it works in closer related species like mouse and rats, but there are several groups working on that. And it could, of course, minimize the rejection rate, but you have to take into account that the vessels still come from the host, still come from the pig. So you would need some approach which uses genome editing of the pig and yeah, the injection of ES cells. And at the end, the question is if you can produce a lot of pigs with that technology, because you cannot breed them, then they would not produce a human organ. You cannot clone them, they would not produce a human organ. So you would always have to inject these human cells into zygotes that have a deletion of a gene that is important for organ formation. But I think Banu is also in some of these projects that are working on chimerism.
Thank you, Bjorn, for your excellent presentation and the answering of the questions, some of the questions. We have the privilege to you and then for accepting our invitation and the sharing the research. Please answer your slide because the next presentation we will have from Dr. Bhanu.
Right. It was my pleasure. It was my privilege to give a talk here at your nice symposium. So stop. So I stopped it.
Yeah. Hello, Dr. Bhanu, you are around? Hello.
Yes, I'm here.
Okay. Next presentation, we have very eminent scientist. He is basically from India. He is currently the associate professor in the Division of Animal Science at University of Missouri. Dr. Telugu, he is also a co-founder of the Ren Ovet Bioscience, the livestock genetic engineering company with primary working in Maryland. And primary research interest of the Dr. Bhanu Lab and his company is the genetic engineering and biotechnology. Genetic engineering, biotechnology, and reproductive biology, particularly for farm animals. The laboratory employs the genome editing tools, particularly CRISPR-Cas, for site-specific altering of the genome in large animals such as the pigs for agriculture and biomedical application. Specially use the genome editing tools to employ the alleles to facilitate the rationale selection for agricultural traits. The laboratory is actively engaged in developing porcine model for human diseases, for example, the diabetes, cardiovascular disease, and the obesity, and developing technologies for the transplantation research where the pig is preferred as animal model. His area of specialization, as I mentioned, is the animal biotechnology, reproductive biology. As of more than the 45 publications and the total citations, more than 2,037 H-index. He has been recipient of various awards, for example, Bill and Melinda Gates Awards, Vision Grant Awards, Junior Faculty Awards, and Invention of the Year Award, and Inventor Pitch Award. A major achievement of his research was the first to develop the technology for targeted genome editing in livestock zygote. He's currently using the genome editing and base editing. Several projects he has been working and several students he has mentored. So now I invite the Dr. Bhanu for his talk. Dr. Bhanu, please. Hello. Yeah.
Yes. Let's see. Can you see this all right?
Yeah. The slides are visible. Right. Voice is clearly audible. Please.
All right. Thank you very much for the kind introduction, Naresh and the organizers. It's a privilege to be here and talking to all of you. It's really a tough act to follow after Bjorn's presentation. He just blew us away with all his work in agriculture and biomedical work. Today, I just want to focus on some of our work as it relates to precision breeding in agriculture for agricultural applications using genome editors. So the primary research interests in our laboratory are using genetic engineering for both agricultural applications as well as biomedical research. From an agriculture standpoint, I'll be sharing a few projects with you today. We're interested in bringing in genome editing solutions to improve animal welfare, animal well-being. So this may be improving disease resistance and so forth. And finally, being able to solve the food security issues. So for these particular interests, we study in pigs and also in cattle. From the biomedical standpoint, as Dr. Naresh pointed out, we use different models. So we work with mouse models, we work with pig models, and we also work with human stem cells. We use them for disease modeling, and we also study for regenerative medicine applications. And as you're aware, there's also a cross-section, which is One Health. So diseases that affect pigs, that are zoonotic, can also affect humans. So we have some interest in using genetic engineering for One Health applications. So, with that said, this is the broader outline of my presentation today. I'd like to start off by asking the question, why perform genome editing in livestock species? So in the context of India, so I'm specifically asking the question, how can we bring genome editing solutions for agricultural breeding programs? I'll provide a few case studies in which we have utilized them for bringing in solutions for agricultural applications in both cattle and pig, and I'll conclude the talk. So I would argue that precision breeding is here. It is latest in the evolution of various breeding strategies. As you're aware, breeding, the end goal of all the breeding applications is bringing about genetic change. So in the beginning, we started with mass selection. So over the millennia, we've been doing this. So the idea of changing the genes or manipulating the genome is not a novel concept. We've been doing through centuries. So the way we do it is actually improving along the way. We believe that the genome editing or precision breeding is the latest tool in this long scheme of breeding programs. So like I said, the way we're doing breeding and bringing about genetic change is differing. So we started with mass selection. So we used to take a genetically outlier male, and we used to take a female that looks phenotypically distinct, and we used to breed them. So there's a lot of guesswork involved there. But as we went forward with the utilization of the genetic tools, now the tools have become much more refined. So then came the pedigree selection. So we could see how the progeny is performing. With the advent of genome sequencing, we are moving on to market-assisted selection. In the 80s is the first time in which we were able to bring in the genes from outside of your species. So I argue still that transgenic is the singular most powerful breeding tool because it allows us to bring the traits that are not available in your native species. With the sequencing of the genomes, now we could do genome-wide selection. So by the time the offspring is born, you can reasonably predict the performance of these animals. So you don't have to keep these animals for a very long period of time, and not have to worry about the cost in selection of these sires. So with the advent of genome editing, now we have entered the era of precision breeding. So we not only can predict the success of these markers, but we can now have the ability to move these across different species, and even within the species across different breeds. So again, I still didn't explain why we should be considering genome editing or how this would be an advantage over the traditional breeding process. So the downside to the current breeding strategy is, we're all familiar, it is a very slow process. It takes about 5 generations of breeding and backcrossing to basically get 30% increase in genetic performance across animals. And when you consider that as the case example of cattle, that would be close to 30 years. So it is a very slow and steady process. So it is very non-responsive in my mind. So for a mean generation interval, like I was mentioning, a 30% could be about 10 years. And the difficulty with this, again, is low heritability of traits. So even if you have identified a genetic marker that provides, for example, disease resistance or improves milk production, the heritability of that trait could be limited when you're breeding that animal. So I'm just going to use 2 examples with 2 haplotypes here. So haplotypes are segments of the chromosome that are inherited as a unit when you're breeding an animal for successful generation. So consider 2 haplotypes here. The one in the red is the allele that you want to transfer to the next generation. And consider in a scenario wherein this beneficial allele also carries another undesirable allele on the same haplotype. And on a second haplotype, you have a desired QTN that, say, improves the milk production. And if you want to use this in a breeding program, you can see that all of the 4 different gametes would have different combination of these 2 haplotypes. Only 2 of your gametes will have the desired allele. Only one of the 4 gametes that is going to be produced will have the combination of your desired QTN, desired allele, and also the desired QTN. So you can see the heritability of these traits from this animal just with 2 haplotypes is already 1 in 4. And if you throw in different combinations of haplotypes across the genome, you can see the chance of you getting that rare haplotype combination passed on to the next generation is very limited. So the inherent bottleneck with the traditional breeding programs is this the low heritability of traits. Even in this best case scenario, you may still end up with a desired allele, a desired QTN, but you're also bringing with you the undesired allele with it. So this brings about what we call as a loss of other valuable traits. We call it a Hitchhiker effect. So this allele that is passed on with your favorable allele. So every time you're selecting for this, you're also bringing in this undesired allele. So this is very evident whenever you're strongly selecting for a particular trait. So this is an example for a dairy cattle. So in the United States, it's a great success story. You have seen that in the last 50 years or so, the milk production has actually improved 100%. So we went from somewhere between 13,000 pounds of milk per animal to about 26,000 pounds. It's a remarkable success story. This is done with just selection, traditional breeding program, improved management practices, better nutrition, and so forth. So here we did not use any genome editing or any transgenics. But what you have seen is that in the period where we are seeing improvement in milk production, you see a simultaneous decline in reproductive performance within these animals. So again, this kind of strongly suggests that a singular focus on a production trait will result in loss of other traits that may be of value to you. So the singular major contributor is this Hitchhiker effect, as I was mentioning to you. And So this is example I was mentioning to you. So what we are arguing is that genome editing could be a possible solution to overcome both of these limitations. So one is low heritability, and also maybe also able to eliminating the hitchhiker effect. So you can go with editors like Bjorn has mentioned earlier, take out that undesired allele that always tags your beneficial allele on your desired haplotype. You can also do replacement so that in one generation, you actually have improving the chances of getting your desired alleles, also in combination with our desired QTNs. Another downside to the current breeding strategy is that your beneficial trait needs to be present in the population. Like Bjorn has mentioned earlier, African swine fever is endemic to warthogs. The warthogs can get the disease, their natural host. It doesn't do anything to them. But if the same virus crosses over into the domestic pigs, it's absolutely devastating. The pigs will get hemorrhagic disease and they'll die in no time. So just the numbers of the pigs that had to be euthanized, 150 million or so in China, like he's mentioning, is mind-boggling. So if you want to bring in these traits that are existing in the nature into your population, our current management or current breeding strategies would not be conducive, because you cannot imagine a scenario in which you can breed a domestic pig with a warthog. So we need to have the ability to bring these favorable alleles, not only across these animals into our native animals, to as a tool for improving their performance overall. And again, that is not conducive or favorable with our current breeding strategy. So what we're arguing is that genome editing or genetic engineering is a method by which we can leverage this. So we should be able to harness the wealth of genetic markers and alleles that are present in our population, and being able to bring into our breeding programs. And we can do so seamlessly by identifying this and using a genetic engineering strategy to just modify the targeted areas, and you could accelerate the genetic progress within our current livestock systems. So with that brief introduction, I would like to lay the groundwork in which we have done exactly that. So we have identified traits that are not present in our population, and we are figuring out a way to seamlessly introgress those favorable alleles into our livestock population. So one such example is, we recently published this article, go into some depth, but we are introducing, we have, as a proof of concept, we have asked the question if you can bring in novel prion variants that are existing of all things human, that we know are provides resistance to the prion disease, and using that as a mechanism to generate the mad cow resistant cattle. So the objective of the study is, first and foremost, is to optimize the reproductive technologies. So this is the first for us in our lab. So we wanted to make sure that we can get optimal reproductive performance, that we could standardize the genome editing approaches, and we're asking the question whether we can introgress novel traits into the population. The rationale for the study, as I may have briefly mentioned, prion knockout animals are viable. So even errors at that targeted mutation site in the prion gene would be tolerated in these animals, and we will end up with live animals. And a natural variant occurs in humans that we have identified that provides resilience to the prion diseases. So we wanted to see if the allele that is present in the humans that makes them resistant to prion diseases could be brought into cattle, because cattle are still the natural host for the disease. So a brief background: prion disease is misfolding of the prion proteins. There are several manifestations of this disease. In humans, we called it, they have different names, such as CJD, fatal familial insomnia, Kuru. In cow, we called it mad cow, but it's a encephaly, spongiform encephalopathy. In sheep or goats, we call it scrapie, and we also see in other ruminants like deer and moose, it's a chronic wasting disease. We first stumbled across this article that came out in Nature, where they identified a naturally occurring variant of the human prion gene that exists in this native population of humans in Papua New Guinea. So I'm not going to go into too many details, but as a population that practices cannibalism, there was an epidemic of prion disease called Kuru within this region, and these people have been identified to be resistant to the prion diseases. And the major change that these guys inherited that provided them resistance is this modification of glycine to a valine within the prion gene at a specific site that allowed them to be resistant to misfolding and also development of the prion diseases. When this particular variant was made as a transgenic and introduced into the mouse, which is actually not a natural host for the prion diseases, so they knocked out the endogenous prion gene and introduced this variant within this mouse as a transgene. They were able to see that this particular prion is actually, these mice are resistant to all 18 known variants of prion proteins. So clearly, this variant that has been selected within this population provides greater resilience to the development of prion diseases. So we wanted to ask if we could do the same thing in cattle, unlike mice, which is actually a natural host for this disease. So in the process, make these animals resistant to various prion diseases. I'm going to go into some detail. So the amino acid that we want to change, the glycine to a valine, is located here. What we're trying to do is change these two nucleotides, GC, to a TA. And that's about the only change we want to do in that entire genome. And the expectation is that now you would have inherited the variant that is not present in our cattle population, but now they would have achieved the same level of resistance that these humans have exhibited and the mice have shown in experimental studies. We tested 2 different CRISPR guide RNAs, and our objective is to do the genome editing directly in the embryos. This becomes very important, especially in cattle and in also the production schemes of various livestock genetics companies. They don't want to do cloning. They much rather do genetic modification directly in the embryos. One, the fertility rate would be higher, and 2, like Dr. Peterson was mentioning earlier, you're not going to deal with associated problems that come with somatic cell nuclear transfer cloning. So for the context like India, this also is really important because we would like to be able to just collect some embryos from your breed of interest and be about and go and do the genetic modification directly in the embryos. So you're maintaining the genetic merit, you're not losing offspring through cloning associated problems, and you'll be able to affect the genetic change. So we were very determined to make this work. This is not a trivial task because you are racing against the CRISPRs who are cutting at that site. So you would need to have an optimal cutting as well as replacement of these new alleles at that site. So we then went ahead and systematically did screen all these guides and the targeting vectors. I'm not going to go into too much details, but this is a priority when you're working with animals with long gestation intervals. You cannot just hope for the best, right? So you're looking at 9 months of pregnancy gestation time. So in this case, we had tested these guides, CRISPR guides. There's a forward guide, there's a reverse guide, is material here, but we have seen that not all guides are equal. There is this reverse guide, which is in the orientation and is close to this modification, gives us better targeting efficiencies than the guide that's cutting outside of the target site. And we have tested numerous embryos over multiple replicates, and we have clearly seen that this particular guide works better. Then we have tested the combination of the oligos of the single-stranded DNA that would be used to introduce the change. Again, targeting this strand versus the antisense strand, and the antisense strand seems to work better. And then we wanted to go a little bit further and look at the level of mosaicism that comes with these approaches. So whenever you're injecting into a single cell embryo, for each subsequent cell division, each of these embryonic cells would have accumulated mutations that are not intended. So you would end up with an animal with a host of different modifications. So in our case, we utilized Illumina's iSeq. We were doing targeted amplicon sequencing. So this is, assuming this is the genetic site you're modifying, you would use primers across this, using the barcodes, you multiplex and do multiple reads for each animal. So in this case, for example, each of these are individual embryos. We are sequencing up to about 3,000 reads for each embryo. So that gives us in-depth analysis, at least at a 10x coverage. So assuming there are 100 cells, 200 copies of genome, you need about 2,000 reads to get about 10-fold coverage across that cut site. So we were able to look at least a 10-fold coverage across all these embryos. As expected, the wild type locus is shown in the top, and this is the modification we want to achieve. We've seen anything from 10% or 3% all the way to 85%. So long story short, the HDR targeting approaches seem to be working, but it's also highly variable. So you can go with very little to a lot of modifications. So moving further, tested various embryos, various combinations of them, and finally settled on, we tested 3 different oligo combinations, and we finally settled on this particular set of guide RNA / targeting vector combination that has given us pretty much consistently greater than 50% of the targeted allele being more than HDR or targeted modifications at the site. This is important because it ensures that at least in the subsequent generation, half of your offspring would have inherited the targeted modification. From a round of embryo transfer session, we did embryo transfers into 12 animals. 7 of them went to term, and we got 6 live edited calves and 1 wild type control. Wanted to test our embryo transfer efficiencies. These are the animals that were born. These are all slaughterhouse-derived oocytes, so you can see the genetics not being equal among all of these. We further went and analyzed these targeted frequency within these edited calves. So for each of these, we collected samples from the ear or samples from the blood, and this is the reference wild type animal that we have. So the offspring with wild type animals, you expect most of these reads to be unmodified or wild type. Whenever you're doing injections with the CRISPRs, you expect some of them to have just the mutations. So these are other modifications. You would probably see non-homologous end joining based repair mechanisms, and you expect to see homology directed repair. What we have identified, rather we're pleased with it, is that we saw consistently among 6 offspring, 2 of them actually have greater than 80% HDR frequencies. This particular animal has about 97% of them as HDR, non-homologous end joining. This one has about 50% NHEG and HDR, and these 2 animals have more than 95% of the reads representative of the targeted genetic modification. So we may actually have generated an animal that's close to 100% targeted modification at the targeted site. And this is representative in both the ear sample and as well as genomic DNA from the blood as well. We euthanized one of the calves to look at the mosaicism. So we collected various tissues, collected the genomic DNA, and we ran the same analysis all over again. And what is seen with the blood and the ear is representative across various organs as well. So clearly showing that the frequencies that we are seeing are representative across various tissues, and is likely going to be transmitted at a higher rate. What we were surprised to see is that 90% somatic mutation rate is not transferring at the same frequency. So when we collected the sperm and we then did the sequencing, that rate fell to about 57% or 60%. So regardless, I believe that this is a first proof of concept where we have shown that you can bring a allele which is favorable, that provides disease resistance, that can be brought across species barriers into your breed of interest. And that it can achieve close to 50% of the targeted modification in one generation. Clearly, these efficiencies will need to be improved, but we're very pleased with with this preliminary attempt. So this could be a precursor or at least a palette template for doing precision breeding in our endogenous cattle as well and buffaloes. So the ability to do this genetic modification directly in the embryos allows for scalable genome editing solutions. So you can just go into your favorite breed, you can get the embryos, harvest the embryos, deliver your combination of CRISPRs with a template, transfer into animals that doesn't have to be of superior genetic merit, and be able to have the edited calves within one generation. Because you're doing in embryos, you can actually really scale this up real quickly and identify the founders that have the highest targeted mutation rates, so that can be employed into a breeding program quite effectively. All right. So with this basic example, we're working on other traits that would be of greater interest to our Indian subcontinent. So this is an example in which we are trying to make cattle that would be more heat tolerant. This publication came out in 2014 from a group in New Zealand, where they identified a mutation in the prolactin receptor that introduced a stop codon. So the net result is you have animals which are naturally showing this phenotype that have very thin coat, so they have very slick hair, and the sweat glands are more pronounced. So the expectation is with the less fur coat and actually better perspiration rates, they actually have been shown to be more thermoneutral under heat stress conditions than the wild type animals. So we're trying to bring this trait into our native cattle in Seminoles, from Seminoles to our Angus and other native cattle. So we're currently working on this project with funding from the Gates Foundation. So the expected phenotype from this, like I mentioned, it has short, slick coats. They have improved heat tolerance. And the upside with these animals also, because it involves prolactin receptor, is that it improves the milk production in these animals as well. There are other traits that we're working. Similarly, one is a prolactin receptor I mentioned to you, one growth hormone receptor. So as you're familiar with, Monsanto had a product called Posilac, which is actually growth hormone that improves milk production. We have identified a mutation in the growth hormone receptor that brings about a similar anabolic effect, and we're trying to bring them back into our cattle. There are other modifications that we're currently working on. One is the DGAT1 that improves the fat percentage within the milk. Typically, that improves the value of the milk that is being produced. And another one is the ABCG2, which is a transporter protein. So in all, we're trying to bring genome editing for the precision breeding applications in our livestock. And at this current moment, we're working on heat tolerance, disease resistance, and improving the production traits. So significance of the study so far, we have shown the feasibility of introducing targeted genetic modifications and integration of novel variants by direct injection of editing reagents into the zygotes. We have successfully validated that and are currently employing these solutions in our system. I'm going to switch gears a little bit and switch species. I'm going to talk about a second project in which we are trying to do the similar thing. We're trying to bring variants from outside of the population into our animals. So in this case, we are trying to see if we could bring novel variants that are found in rat that can be employed into our pig breeding programs for elimination of the boar taint. Dr. Peterson gave a good segue for my talk here. He mentioned boar taint briefly. So I'm not going to go into too many details, but the objective of the study is to use a genetic solution to eliminate boar taints in pig using genome editors. Again, as a backdrop, rats do not exhibit taint that comes with the steroids. And our question is to see if we could learn what is present in rats and maybe deploy them into the pigs, which actually have the problem. And the rationale for the study, as was mentioned by a previous speaker, boar taint is an offensive odor that emanates from uncastrated animals. It would require physical castration, and the current solutions like vaccinations have either failed or they're too expensive. This is a brief overview. We don't need to go into too many details, but the main culprit for boar taint is the steroids called 16 androstenone steroids. So an accumulation of these steroids, it is again one half of the problem, scatol being the other. The accumulation of this is the main problem that results in boar taint. So the enzymes that are mainly involved, or the steroidogenic enzymes that are mainly involved in this process, are these two enzymes here, CB5 and CYP17. And you cannot just go ahead and knock these out because they're also are important for producing testosterone. So in this particular case, a knockout strategy is not relevant. We need to basically see if we can modify the steroid enzymes so that we have a phenotype that most closely mirrors the rats than that of the pigs. There is some genetic precedent to it. We have identified rare polymorphism in the pig CB5 gene that actually causes the reduction in its production and decreases the synthesis of androstenones. CB5, like I mentioned, you can knock it out, but it also knocks out other androgens as well. So we cannot just go ahead and knock it out. So here we employed the comparative genomics approaches. We looked at the steroid binding pockets of these two enzymes, CB5 and CYP17. I'm showing the sequence that exists in rats on the top and what's present in the pig. You can see there are key changes in amino acids within the steroid binding pocket in the case of pig that is not present in the rat. And so is the case with CYP17A1. Again, the amino acids present in human and pig are different from the rat in the steroid binding pocket. So long story short, what we would like to do is bring these modifications into our pigs at that particular site, and we hope that would bring about the solution. And from a preliminary in vitro screen, we did a large scale genetic modifications of these two enzymes and performed steroid assays. And we actually were able to find a combination of these modifications within the steroid binding pockets of these two genes that has resulted in a decreased production of androstenones. So we went ahead and started making these pigs. And you know, at this point, this doesn't mean anything, but I just want you to show that we are successful in bringing about this genetic modification in the pigs. So right now we have edited these alleles and we are making these animals, and we'll be screening their steroid profile at both weaning and puberty to see that, again, as a proof of concept that we can bring variants that are not present in your population from outside and bring about the intended genetic change, phenotypic change. And the last project, which is somewhat a segue for what Dr. Peterson has mentioned, is utilizing these genome editing tools for applied technologies. So I just want to briefly discuss a project. We're currently working on a variation of this, but the idea is that you can actually make animals that could be used as surrogate sires for your elite animals. So the main objective of this study, which has now been published in both PNAS and scientific reports, is to generate males that have germline modifications. So again, with the end goal that we could use these males for transplantation of the spermatogonial stem cells from your intended donor for propagation. So here is an illustration. What we would like to see is generate a cohort of pigs that have mutations in a key gene that would not allow them to produce their spermatozoa. So they have all the testicular somatic cells, but they would not have any germ cells. And with the idea that we can then take spermatogonial stem cells from an elite boar, and we can then populate these animals And even when this animal is dead, we expect robust spermatogenesis from these surrogate sires. So these animals would be reproducing the sperm of this animal, even when we have lost this. So the key advantage here is that a genetically outlier male or a genome edited founder, that potential would be lost when the animal is dead. So now we can not only scale the production of these spermatozoa, but we can keep the genetics alive even in posterity. And on the flip side, you can also see a scenario where this could be a mechanism to amplify the genetic selection. So now instead of having one sire, you would end up with multiple sires that can be utilized for amplifying the genetics in your breeding programs. So you can easily replace this with a cattle and your favorite bulls. And you can see how you're not limited to ejaculation from a single bull at one time. You can have different bulls being made and could be brought into different sperm collection centers within India so that you can actually disseminate the genetics at a much faster rate. The goal here, again, you need to make the spermatogenesis stem cell knockout pigs. The goal here of this study is to target this gene called NANOS2. We have done embryo zygotic injections and performed embryo transfers. We identified that all piglets from this study, about 15 of them are edited. In those animals that have true knockout for NANOS2, we could see that the spermatogenesis stem cell seminiferous tubules are intact. You have the Sertoli cells that are more or less intact, but there is no spermatogenesis stem cells, or there is no evidence of spermatogenesis, as we have seen in the heterozygous animals. And you can see in the knockouts, these seminiferous tubules are more or less empty. When we ejaculated these animals, we saw robust spermatogenesis from heterozygous knockouts, but there is no evidence of spermatogenesis in NANOS2 null animals. The Leydig the supporting cells, both seminiferous tubules are intact, but also the Leydig cell functionality is also intact. The testosterone production is normal within these animals. And on the flip side, as is expected, knockout of NANOS2 does not result in loss of oogenesis in the females. So you can keep this knockout in the female background and use the heterozygous animals to breed them. So in the next generation, you would end up with half of your boars being knockout for NANOS2. So you can also amplify the genetics easily. So in summary, we have identified that the role of NANOS2 is conserved in pigs, as is other livestock species, which is confirmed by now. Males and females that are heterozygous are fertile. They produce litters. Female knockout animals are also fertile. And the future goal is, with our collaborator at Washington State University, Dr. John Oatley, we are standardizing procedures for SSC transplantation. So with this, I'd like to conclude that, and that hopefully convinced you that precision breeding should be considered as a solution in our breeding programs. And they have several advantages. Chief among them are there's increased precision and efficiency of introducing desirable traits. Conventional breeding is a random process. As I mentioned earlier, with the various combinations of haplotypes, it's like a genetic lottery. You may or may not get it. But with precision breeding, there is a greater likelihood that that particular modification would be transmitted to the next generation. It increases the heritability of traits. We have the ability to now separate linked genes, eliminate the Hitchhiker effect. So we can go into the population not only to improve the alleles, but also get rid of these undesirable alleles or traits. And the single greatest advantage is our ability to bring in novel traits into our population, which is not possible with conventional breeding. So with that, I'd like to acknowledge the team that does bulk of the work, and our funding sources and collaborators. That I'll take any questions you may have.
Yeah, thank you, Dr. Banu, for the excellent talk, and your share these novel ideas for making a disease resistance large domestic animal like the cattle, and maybe apply that knowledge to the buffalo too in India. Milk yield and fat percentage to modulate to how can improve the milk yield or change the fat content in the milk, and heat tolerance animals. And the last we have been discussed the pig, the surrogate side technology will be useful. And if is there any questions in the Q&A box? There are a few questions. The first question is from Ravindra Kumar. Does the genome editing affect other fitness trait? And is there any study on this? The fitness trait he was asking.
Yeah,
Hello.
so yeah, that is a very good question. So one of the greatest concerns with all of the genome editing solution is potential off targets and potential unintended consequences that may potentially come with this. So all I can say is that we are currently working with an industry partner, Genus PLC. They are working on these solutions to bring them to breeding programs, and we are doing large. So in a nutshell, you need to be able to sequence the genome at a greater length to see any unintended consequences, and whether if they would, those changes would affect your performance as well. We have seen that you could actually improve the likelihood of on-target and decrease the likelihood of off-target effects by spending some time up, in the front, optimizing the reagents. So in our example, where we did the cattle project, we spent almost a year optimizing the reagents before we went and did an embryo transfer experiment. So there needs to be a little bit of more jurisprudence when working with these livestock species, because the pregnancy interval or gestation interval is too long.
Okay.
But that's a good question, and will be,
okay.
We'll know in the years to come.
Yeah, thank you, Dr. Banu. There's one question from my side, because you have been used the micro-injection for production of the treated animals instead of the SCNT. There are some limitations with SCNT, but if there is any mosaic animals been produced, and what percentage you have been observed in your experiment that you get the mosaic animals percentage in terms of the micro injections, and how can you then manage that over these SCNT?
Yeah, so just so you know, in our lab we also do SCNT. So I'm not against SCNT. So I see a value in using that. Like Dr. Peterson has mentioned, you would like to make a cohort of animals that have the same mutation in the same genetic background. So it makes it very easy to just have clonal lines of pigs on a wild type and a knockout to study those. So we do SCNT a lot. But from a production standpoint, especially if you were to make these animals here in the US, the producers don't want to do SCNT because that's another trigger for FDA process regulation. So they would much rather go and do this directly in the embryos. And that's why this study, I believe, is important. And then in regards to your mosaicism, we see quite a bit. So I'm just going back to this published study that I presented. So again, these are different offspring from that one embryo transfer study, right? And you see, a few animals that have only the non-homologous. This is not what we wanted. This is, unintended modifications there. This particular animal has about 50% HDR. Again, this animal, is not very useful to us, but you have offspring that have, very high, almost 95% HDR frequencies. So the goal should be, now that you have preliminary proof of concept, now we are trying to see if we could improve those percentages more reliably in the offspring. So it may, in the end, it come down to optimizing your editors to improve the HDR frequencies. It's a very tricky process because you have to optimize the timing, dosage, and you need to get everything right within the embryo to improve this. So that's an ongoing project in our lab to see how we can improve the HDR frequencies in the animals. But we're pleased to see from preliminary trial that we're getting reasonable hits.
Yeah, thank you. Does you have got the prolactin mutations you have been trying to do for increase the milk yield? This is studies ongoing, or you got some encouraging results?
So we already made the calves, so they're born.
Okay.
So we have live calves on ground. So again, at this point, we need to, let them go to puberty and then breed them to study the phenotype. So that's an ongoing study.
Yeah, that is particularly milk fat and milk yield is really exciting, particularly for the Indian context, because in India the milk has been based upon the fat percentage. So
right.
When you got some results, we will go through your publications and your research, and that will maybe some useful for the Indian scientists. Thank you, Dr. Bhanu, for your exciting talk and for sharing your research. Thank you.
In closing, I'd just like to thank you for the invitation, but I'd like to I just want to mention that we'll be open to collaboration, especially working with buffaloes. We have been invited to present concept notes for the Gates Foundation. So if any of you are interested in collaborating, we may be able to get some funding as well.
Yeah, sure. I think we are happy to contribute, or we are happy to collaborate with you. And because it's really exciting, the research we want to also explore in buffalo too. There is one more question from the Carlos Joan. In your opinion, what would be the main advantage of using the surrogate sire through spermatogenous stem cell transplantation instead of cloning as a tool to replicate valuable sire? In the same way, what I think you respond, Dr. Bhanu, please.
Yeah. So in the genetic amplification schemes, so that's really where we're coming with this, that cloning is not a solution because we don't know what the epigenetic modification that bring with them as well. So that is one concern. Maybe it's not a huge concern. Second thing is there may be advantages in some of these breeds that are smaller breeds, or for example goats, small ruminants, where we have not adapted this sperm collection or the dissemination of the sperm as we have done with the cattle, right? So animals that are smaller, Punganore cattle, for example, so the ability to collect sperm, freeze them, and if you have a genetic outlier male, being able to store that genetic value for subsequent generations, we believe there is an advantage to tap into the surrogate sires where you can just harvest the spermatogenetic stem cells, and that would be, in theory, would be able to produce robust spermatogenesis in the resulting offspring. And you could have multiple nucleation centers across India where you could just do this surrogate sires, so you can readily disseminate them at a higher frequency. So if you have a genetic outlier male, we argue that this could be a mechanism to amplify the genetics and disseminate them in your target population. At least that's one major application that we're thinking about. We are also working on an alternative where we could do this from the female side as well. So you could do both surrogate sires and surrogate dams as well. So hopefully in the future, we could share the results with you.
Yeah, thank you, Dr. Bhanu. Now I hand over to Dr. Manoj.
Okay, thank you, Dr. Naresh. Now we are moving to our third lecture by Dr. Naresh Solekar. So Dr. Solekar is scientist at National Dairy Research Institute, Karnal, India. He was instrumental to establish the simplest handmade cloning technology in buffalo. He worked at ICR Central Research Institute on buffalo, Hisar, and then he shifted to National Dairy Research Institute, Karnal. Dr. Solekar won several awards like DBT, IYBA awards, ICR Interdisciplinary Team Award, NASI awards, and he having several achievements also. He optimized the buffalo cloning technology in India and produced more than 10 clone buffaloes from single elite animals and established a somatic cell bank in case of buffalo. He working in several projects. He having ongoing 5 projects and completed 3 projects. He also guided several events, and he is very young, and he published 32 papers. Okay, so now Dr. Naresh, now I invite you to proceed your lectures, please.
Thank you, Dr. Manoj, for the introduction. That is more nice introduction. I think when the Beyond and the Dr. Bhanu has presented their works on the genome editing, we have been started in the last couple of months back. We initiated the genome editing research, particularly our important dairy animal in India, that is a buffalo. So what I highlighted, the two projects we currently been initiated in India, particularly for the Central Institute, my previous organization where I worked for the 7.5 years. So there I have been initiated when I transferred recently to the National Dairy Research Institute. So I continuing that project at the NDRI, but my talk will be comparing the two parts. One part is considering the what we have been optimized the assisted reproductive technology in the buffalo, particularly animal cloning, and then I will share our attempts in the genome editing, particularly to improve the productivity or disease resistance in the buffalo. So if you look at the buffalo globally, so like the cattle, pigs, and the other domestic animal, the total buffalo population in the world is a 208 million population, after the majority of the 90% have been localized in the Asian countries, primarily in the India, which shares around the 57% of the global buffalo population, then the China and the Pakistan. And if you talked about the contribution of buffalo in terms of the global milk production, 50% of the global milk is produced from the buffalo. And again, in Asia, 35% of the Asian milk produced in the Asia is shared by the buffalo. And if you talk about the India, though the population of buffalo is a half than that of the cattle, so currently the Indian population 109 million buffalo population, which is the half of that of the cattle, but the contribution still is around the 50%. And still, if the milk production, buffalo is not only animal which produce the milk, but is also preferred animal for the meat. So 4.2 million tons of meat produced annually globally, and in India, in every year, so Indian government has been exporting buffalo meat is more than the 20,000 crore rupees. So if you look at the characteristics of the buffalo as they categorize, there are the two type of buffaloes. One is a river buffalo, which the majority of the Indian breeds are belonging to the river buffalo. There's a 50 chromosomes number, and is specifically raised for the dairy breeds. Another, there's a swine type buffaloes, the 48 chromosomes number. The northeastern part of the country, India, there are swine buffaloes, or majorly the swine buffaloes, morely localized in the China, Thailand, and Indonesia. The India and Pakistan, they have these countries have the river type of buffaloes. And the famous breed, I think everybody knows that the Murrah that belongs to Murrah, Jafrabadi that belongs to the India, Ravi that belongs to the Pakistan, which is also the high-eating buffalo, and the Mediterranean buffalo in Italy. So Italy has been has the commercial buffalo has been the practices in the Italy, and still in India we have the household buffalo has been the practices. And if you look at these countries, the Asian countries, particularly India and Pakistan and Bangladesh, this is an important animal for the agriculture and economy. And now there are several herds has been established in European and the Latin American countries. For example, Venezuela, they have these important buffalo, also an important animal in these Latin American countries. So since we have been working from the last couple of years on the somatic cell nuclear transfer, so in India, we have initiated the SCNT in 1996, particularly in buffalo, but that time we did not succeed to produce the live animal. The first clone animal produced in the country in 2009 by the National Dairy Research Institute, and the technique they have established in the country that is for the handmade cloning technique that is a different than that of the micromanipulation technique. So current what they through the last 10 years research, we get the embryo production rate around 30 to 35%, and live birth rate we achieved from the last couple of years 6 to 8%. But still in the SCNT we have some limitations. For example, the pregnancy losses, 25 to 30% pregnancy losses. What are the pregnancy we established? They have been lost in between till the term. So the different types of the cells, when we are optimizing the cloning technique, different types of the cells are used. For example, the skin product, which is most commonly used cells, stem cells, we are able to produce the clone animal from the stem cells, and some of the important particular there is a noticeable examples in the buffalo, that is when the production of the clone from the urine derived cell as well as from the semen derived cell. These are the first reports across the any species that produce the clone animal from these two important non-energy cell source. If you talked about as on 2021, There are the two research organizations. One is NDRI itself and another research organization in India, that is the Central Institute of Research on Buffalo. Both the organizations working on the buffalo cloning technique. And at present, we have the 21 clone buffaloes, of which because currently the Indian government wanted to multiply the elite genetics of the breeding bull. So we have the 16 clone bulls available. The 10 clone bulls are available at the CIRB, my previous organization, and these 6 clone bulls are available at the NDRI. So recently, in the last year, the CRB produced the 7 clone bulls from a single breeding bull. Also, the proof of the principle to reclone the already cloned bulls when the clone bull, that clone bull was born in 2015, and 2019 that clone bull is recloned. Whatever the study, what we have been conducting in the buffalo, to see that whatever the clones we have in both the institution, they have the normal growth, health, fertility, and the production. So we compare the hematological indices, the immunity of these animals, and the fertility and the production. Recently, we published the papers in the scientific report where, as well as in PLOS One, that highlighting that the clone bulls, whatever we have in the country, they have these, those are the maturity, they attain the maturity, they have the normal fertility and the semen parameters. So these are the recent achievements. So these are the pictures from the 7 clones are produced from the single breeding bull, as well as this recloned calf that's produced from the already cloned bull in 2015. These are the achievements from the CIRB in terms of the buffalo cloning. So as when I was talking about the buffalo cloning, this technique of the buffalo cloning is different than that of what has been used in the other countries, but still the handmade cloning is well-established technique in cattle. The China is extensively used the handmade cloning to produce these clone as well as the genome edited or the transgenic animals, as well as some in Australia or some European countries, they also use this handmade cloning. The basic difference between the handmade cloning is that this technology is the free of the micro manipulator. There is no need of the micro manipulator. Only stereo zoom microscope can be used to enucleate oocytes. So this is a major difference. Other technical difference is that these are the embryos are the zona free. These embryos don't have the zona pellucida, so the hatching problem will not be there. So the embryos are already hatched. When they reach to the blastocyst at the 6 days or 7 days, they are already hatched, and then are very easy to transfer in the recipient animal. The technical difference between this traditional SCNT and this handmade cloning is this use of the nuclear cytoplasm. So in case of the handmade cloning, there are the 2 enucleated oocytes are there's a sandwich, and 1 somatic cell are electrofused between the 2 enucleated oocytes. And that against the debatable issues for the concern for the handmade cloning, that there is the chances of the increasing mitochondrial heteroplasmy. Our team, they've been looking into this mitochondrial heteroplasmy in these, what are the clones we have. At present, we have these 16 clones. We are looking at that, is these animals, they have these mitochondria from the one source or the another source. This is all about the animal cloning, because to have this transgenic or the genome edited animal, from the any laboratory, these laboratories should have the well-established protocol of the assisted reproduction, either the microinjection or the in vitro fertilization or the SCND. So in India, both these institutions, CRB and the NDRI, they have this well-established the SCNT as well as in vitro fertilization and also the microinjection too. In buffalo, there are very scanty publications or the research has been done, particularly in terms of the genome editing. So there are some of the publications I summarize here. This is a 18 years, this ribosomal DNA gene has been targeted to the mammary gland. Similarly, the myostatin has been targeted. What the observation was just to optimize the technique and show the proof of principle that this technique has also been worked in buffalo, because very few people are in the world, they have been working and they have been well-established protocol of the assisted reproduction in the buffalo. And that's why there is not many research has been done in case of buffalo. Similarly, from the India, the Paul and the Puneta, they have been worked from the IVRI. They knocking out these carpus luteum function genes. There's a thrombopoietin, TSP1, and EGF, early growth response gene, and how these genes are regulated, these function of the carpus luteum in buffalo. These are some of the reports. What these our laboratory was currently working on this knocking out of the myostatin gene and knocking at the CD18 gene. What the according to the literature which I have searched, at present there is a no live genome edited buffalo produced yet for the any trait. Either it is a knockout, knock-in, or for example the myostatin or any other gene for functional validation. Also, there is no animal is produced yet in case of the buffalo. So what the current project, this project we have initiated in the last year. So this is a multi-institutional project we have been working, the deep center at the NDRI. So here the three species we are targeting, the sheep, goat, and buffalo too, and we targeting the myostatin. Since in India, because the buffalo is not only rare for the milk yield, but also for the meat. Similarly, we have the huge demand of the meat, particularly from the goat and the sheep for in-house consumption. So the considering fact that we proposed the concept to the funding agency, our funding agency ICR, and they considered and they supported us. So what we are targeting, this CRISPR-based approach to knocking out the myostatin and its receptor in the somatic cell as well as in the zygote. Once they have been knocking out, there are a couple of assays, I think, beyond and the Dr. Bhanu or the other previous Dr. Maik or Dr. Wang. They have been already explained a couple of techniques to the presentation. For example, the T7E assay, PCR-based sample sequencing, whole genome sequencing. That's the same thing we are going to do. Once we have the either edited cells or edited embryos after the screening, these embryos will be transferred non-surgically in case of the buffalo, but surgically transferred. There are the specific institution in India. The CRGs work on the goats and the Scary Kashmir universities work on the sheep. And once we able to produce the live animals, the animals, we again need to check that their growth as well as their editing. This is the overall plan, what we are going to do in couple of next 3 years. So why we choose the myostatin? I think both the Wang has been extensively present in knocking out of the myostatin gene. So why we choose these myostatin, or the Dr. Wang or any other people, they choose the myostatin as an important gene, is a unique regulator of the skeletal muscle growth. It is unique, that is a negative regulator. When there is a higher expression of myostatin, muscle growth is down. So the people are trying to knocking out the myostatin so that the muscle yield to be increased. It is highly conserved across the species. For example, it is what we have been analyzed the sequence from the buffalo. It has the similarity with that of the cattle as well as sheep and goat. And this particularly the protein, the myostatin, is expressed in the skeleton muscle. So if you're knocking out the myostatin gene and produce the animal, there should not be technically there not be the large organ size. Basically because the gene are morely expressed in the skeleton muscle. The important characteristic, particularly for the regulatory point of view, because still in India we don't have the regulation how these animals will be treated, but the regulations available in the Europe and worldwide in USA. This particularly these myostatin naturally mutated animals already is available. For example, the Belgian blue or Piedemonte cattle or Texel sheep, and these animals are already in the food chain. So hoping the fact that if you have done these mutations, particularly for the myostatin, these animals could easily pass through the regulatory approval. So that's the considering. And the studies, previous studies, I think Dr. Wang has been extensively describes that either the naturally or artificially mutated myostatin genes grow normal and then fit and they are reproduced. So there is not any abnormality in terms of their fertility fertility index of the animals. So what we have been proceed, so we isolate the genomic DNA from the 2 breeding bulls, because our target to produce the breeding bulls so that the semen can be used for the dissemination if the regulatory agency approves in the future. So considering the fact that we select the breeding bulls, we isolate genomic DNA, we amplify this myostatin exon, we are targeting exon 1, and then send for the Sanger sequencing. Those are the data we got. The ChopChop software is used to designing the CRISPR guides, and then these CRISPR guides are cloned into the PX. There's a 2 vectors. One is the GFP, another one is the furomycin resistance vectors, and these are inserted into this guide. The design guides or procure guides are inserted into these the vectors, and again after insertion, these are again to check that there is the proper insertion or not. These are the basic the technique. Once we have the guide in our hand, what we have been done, because what we observe in case of the buffalo, their buffalo cells are these are very resistant to the transfection. So in first attempt, when we transfect the cells, we note that there is an extremely low expression of the GFP, because these vectors has this larger size. So then we thought that we need to optimize which the conditions need to be done so that we get the maximum transfection rate. We are expecting around the more than the 30% or 40%, around 50%, but that is we could not get that the transfection rate. What currently so for that we use the different conditions, like this is a square pulse has been used, the electroporation, and the different plasmid concentration ranging from 2.5 microgram to 5 and 10 micrograms, and the different electroporation conditions, for example, the 10 microsecond, 1 square pulse, and 300 volt, and what the maximum of these transfection rate we could achieve is the 15%. But with that 15% transfection rate, we could achieve the editing. I will show in my subsequent slide. So this has been optimized in the buffalo cells. And we also, when these buffalo cells are transfected, their dying rate is very, very high. So this is inherent property of the buffalo cells, that they are a little bit difficult to transfect, and there is a specific transfection condition. That's why we did this exercise. And once after we transfect these cells, these are after optimization, which we optimize as a 300 volt, 20 millisecond, 1 square pulse, and the 10 microgram plasmid. We isolate the genomic DNA from this transfected cell. This is a mixed population of cell to make sure that the guides we designing, again, the myostatin gene, they are working or not. And T7E assay is a common method to check immediately that your guide is working or not. If this in the gel picture, if you see here, there is a after heteropelix formation, the process is going through. If there is a heteropelix formation, they will cut into they will give the different bands, so the lower bands, and that indicated that your guide is working and they are doing the editing on the target site. Similarly, we wanted to show how many maximum edits can be done through this. The method is the only 1 or 2 nucleotide change, or there is a large deletion in the sum of the cell. So what we did, we amplify the PCR target PCR fragment and then clone into these TA cloning vector and transform them and pick up the colonies. The different colonies have been picked up as the 10 clone fragments, and then again analyze, and we found that there is a different size is in the edit. For example, 1 nucleotide has been deleted, and maximum we could notice at the 43. But this is the colonies we have been chosen. Maybe the more than that, so we could go up to the 10 colonies only, and that will get these the edits. This indicated that what we got now in our hand, that yes, we have the vector that is working well, despite the fact that we have the lower transfection rate, around the 15%, that works well, and that can be possible to do the editing. The next thing for challenging for us, because at my previous institution, or even at Karnal or at the Hisar, we don't have the cell sorter. So we wanted to go for the another approach. So we wanted to produce the clones from the single cell. So since we have also cloned that same guides into another vector, which is a puromycin, so for that, we wanted to optimize the dose of the puromycin for the killing of the buffalo cells, and allowing after transfection, allowing those transfect cells to be grow to a single cell. So the different doses has been used, and what we have been currently choosing, that is the 1.5 microgram per ml for at least for the 4 or 5 days for the selection into selection of the transfected cell. Another peculiar characteristic of buffalo cells, what we observed during of the limited experiment, but we observed that some of the cells, they are extremely resistant. Even they have these control cells, after you add the puromycin, they are not dying out. So we have to change the doses as well as the time duration of the puromycin when performing the experiment. The next thing, we wanted to optimize the single cell culture. This is again a challenging for us when we think that if we use the transfected cell easily, in some of our 5-6 experiments extremely failed. We could not get any single colonies from the single cell. Then we thought to work on the non-transfected cell first to optimize the protocol, that how we can we succeed that can this buffalo cell will able, because we don't have any information on that. We are following the research protocols or the method that's been available for the cattle and the pigs, majorly cattle and the pigs. But when we use in the buffalo, we will not be succeed in that. So what we have been used the different culture surfaces, media. We also use the conditional media Then we formulated our media, which is based upon this DM and FL, and having the growth factor. The concentration of growth factor also requirement is different in case of the buffalo. Now we at least we have the protocol of the generation of the single cell in terms of the buffalo, which is still we have been low proliferation rate, 20 to 25% only. If you seed the 96 well plate, also the technique we are seeding used for the seeding of the single cell, we individually pick the one cell from the under the stereo zoom microscope, because we are doing the manipulation and the SNT, so we are a little bit more comfortable with the picking up of the single cell instead of the serial dilution. We don't use the serial dilution. We individually pick the single cell, and then we culture them into the 96 well plate, and our these optimized growth factor medium we use, which based upon the DM and F12. We get the proliferation rate. Suppose we seeded 96 well plate, only the 20 to 25 wells have get the proliferated cell. These are the pictures of the single cell proliferation, and the attachment rate also in extremely the poor. It's 30 to 40% only. But is the thing is that it has been possible to generate the single cells in buffalo. This has been done first in case of the non-transfected the cells. In adult cells also, what we observation adult cell proliferation extremely poor. So the fetal fibrillars cells, they have been easy, or the young one fibrillars, they are easy to proliferate and generate the single cell. What we also observe that when we have the 2 or 3 students, they are working, some students get excellent results, some may not be. It's also depend upon how you handle the single cell while the transferring of the media or picking up the cell, which cells you have been picked up. That is also determined that that cell will be attached or proliferated or not. This we did in our buffalo cells. The next thing is that we have earlier worked on the production of the transgenic embryos through the serial dilution method. These are our previous publication. So in which we transfect these the cell, the fibroblast cells, fetal fibroblast cells with the venous reporter, and then use the serial dilution to generate the single cell colonies. When they are transfected, when they are green, they are used to produce these clone embryos. These are the, because as I told you, our embryos are the zona-free. If you see here the picture, these are completely 8 to 16 cell zona-free embryos, and these are the particular pictures of the zona-free embryos. And this is a blastocyst which has been producing the green fluorescence, because we use the donor cell as the green fluorescence. What the method we have been optimized currently to generate the single cell? Through this method, we generate the 5 single cell colonies. That is from the adult animal. These are animals which have been used for the cloning. So we generate from the adult animals to establish and also produce the clone embryos from the 5 established cells, having the integration of the venous gene through the transposomes vectors. And these are the vectors has been provided by the Beyond Laboratory, because myself and one more my colleague, Dr. Dharmendra, we both have been done the postdoctoral training at the Beyond the Laboratory, and Dr. Wilfried and Dr. Neiman. So these people, they are working. So we did this postdoctoral some training in the laboratory. So we haven't got the chances to use these the vectors, and these vectors are also been available in AgG. The next, what we have, when we optimize the protocol for the single cell, then we transfect the cells with the spuromycin vector having the myostatin, the guides against these the guides against the myostatin gene, and then generate the single cell colonies. So through the multiple attempts, we could able to generate the 8 colonies. These are the single cell colonies after the transfection. This is colony 1, 2, 3. And then our next target was to check that these colonies have any kind of the mutations or edit or not. So these single cell colonies are positive. So we picked up once we all get the positive result of the transfected cell with the T7 assay. If you like each band here, if you look at this, particularly the colony number 6, there's the 2 bands. These are the genomic DNA PCR. These are not a T7 assay. These are genomic DNA PCR. From here, the colony number the 8, we have this some smearing. So this indicated that there is some editing, either the monoallelic or biallelic. We wanted to again make sure that what kind of the edit in this the single cell colonies. And these are the pictures of the single cell colony. If you see here, each colony, they have the different morphology. These colonies, for example, they are in flat in shape or very difficult to proliferate, and we could not get the more population of cells. But other colonies, there are some goods, or they are proliferated. Still, there is a mixture of the morphologically. There may be because of the stress of the puromycin or their aging. We don't know. There are several factors that determine the growth of the cells in vitro. So what the next we wanted to the screen that the colonies we have, there is any editing, particularly have they any editing or not. On the PCR, we shows that yes, there are the editings. It's happening, particularly colony number 6 and colony number 8. But there may be possibility that is a 1 or 2 nucleotide cannot be changed. So another protocol that is well established in the Dr. Sachinananda lab, that is SSCP page-based method to quickly screen the colonies that they are editing or not. If you look at in this picture, this colony number 6, there are the different banding patterns. These also indicated that colony number 6 and colony number 8. If you look at the colony number 4, there is a similar pattern to the control animal. So these are some of the pictures of the page assay to quickly screen that these colonies have the specific editing or not. Then what we have done, we have now the colonies in our hand. We wanted to make sure that the colonies have the specific edits or not. So then we again amplify this target region. We clone them into the TA cloning vector, transform it, and pick up these 5 or 6 colonies randomly from the plate and send for the sequencing. And then we got the sequencing results, as we expected, as shown in the band, the PCR band or T7E assay, as well as in case of the page assay. We know the colony number 8 has this monoallelic editing. That means one allele has been knocking out of these 8 nucleotides. So similarly, for example, the colony 1, 2, 3, 4, they don't have any edit. So these colonies will be discarded, and what we have been using the generated colonies, either there's a monoallelic, for example, the colony number 8 and colony number 7, they are the monoallelic in colony number 7, there are the 3 edits has been changed. It's a monoallelic. So we will continue with this generated colonies, particularly colony 6, colony number 8 and colony number 7. And particularly for the colony number 8, we have the biallelic editing. We observe one allele has been edited for the 7 nucleotide and another allele has been edited for the 3 nucleotide. So what we have currently in our laboratory is that we have the 3 colonies that has been having edited at the myostatin gene, particularly one colony as the biallelic editing, another 2 colonies are the monoallelic editing. So these colonies are going to be used for the SCNT application because we are using the cloning method to produce these animals. Because this method is well established for us, so we hope that we could able to produce some edited animals. So our current work is ongoing. On this project, we are working on the screening of the colonies. We have the genomic DNA of the off-target screening of this positive cell line. So because we choose the only 3 the colonies which are the positive edits, and once we have been, and also this technique will explore to produce the more particularly elite animal, either the milk yield, high producing milk yield, or the elite breeding bulls, which is a progeny testing program. So we will choose some bulls, and whatever the technique we optimize till today. We have been working from last 1 year only on this project, so we are very limited the research. But we will try to explore the methods which have been optimized till today to the elite animals, particularly males and the breeding bulls. As I mentioned, these cells will be going to use to produce the clone embryos. Similarly, as suggested by the expert committee, we have planned already that RNP complexes to the micro injection, the embryos. I think the Beyond as well as the Bhanu has already explained that they have been using the micro injection to the zygote. That is to avoid the complications of the SCNT. So what we have currently in our hand, that's RNP complexes with us. So in the next breeding season, in buffalo is our breeding season, so we will try to optimize this micro injection experiment in the buffalo. And then these embryos, whatever the embryos we produce, either through the SCNT or the pronuclear, the zygote injection, they will transfer in the buffalo to produce. This is what we have been achieved still in our laboratory. This data is particularly from the CIRB. And another project we initiated, and one of my students is already working on this project. He is a student, and it was in collaboration between the University of Texas and AM. So in this project, what we have been targeting is that the production of bovine respiratory diseases. This is the Manimia hemolytica infection. The publication is published in 2016 and PNS. It has been shown that the leukotoxins is produced by Manimia hemolytica bind to the intact signal peptide of the CD18 gene. So because in ruminants, either in the cattle, we also in buffalo, we checked that CD18 gene sequence, and we also found that similarly to the cattle. And in this sequence CD18 gene, there is a glutamic acid, while in case of the non-ruminant, there is a glycine, and because of that, the signal peptide, particularly for CD18, is not released from the leukocyte, and they are intact on the leukocyte. So what we have been thinking, this is already, the information is available, that particular screening of the ruminant, except the buffalo, they have also been predicted. So in that situation, we have think that can be used to CRISPR tool to replace particularly the glutamic acid as -5 position with the glycine, because the glycine is available another ruminant animal, so signal vector CD18 in buffalo, if you are able to produce, because in anemia hemolytic infection, for example, there is a similar groups to the HS. So if there is an infection to the buffalo, this is very deadly disease in buffalo too, for example, similar to the cattle. So the animals easily succumbs to the infection, and there is a leukotoxin secreted by this bacteria, anemia hemolytica, and then will affect the leukolysis, and the lung damage in the animal is easily died. So what we have been, again, we have been optimizing the things. What we have been achieved till today, we have the screen the guide, 6 guides that we're designing again CD18 of the buffalo. These are the clones are available, then transfection optimized, and then we screen that T7E assay, we got it, that these vectors are working. We have these donor template to transfect it, but still we at this stage, we are at the cell manipulation, so that to make sure that the changes we are expecting that has been done. This is these 2 projects I would like to share during this what we have been working from the last 1 year, and hope we will collaborate with some more the people, for example, Bhanu or Beyond, or so any other one. We are happy to collaborate, particularly interested to the buffalo as well as the dairy animals, the cattle and the goat. What the sum up of my talk or my of talk is this: we are working on this the myostatin of the knocking out the CD18 gene under progress. So I think the summary of the entire symposium, the 2 days, the talks from the Mike, Wang, Beyond, and the Bhanu, is that any trait can be edited through the CRISPR technology. Either is a production-related trait, or it's related to the health. But thing is that we need to understand that the gene which are responsible for that need to be identified. And that, so what I would like to share with the others, that we have well-established technology, buffalo cloning, only need to have to optimize the genome editing and transgenic technology to produce some of the more model animals, particularly for the buffalo. And obviously, we have the higher opportunities in the country, but in addition to that, we have the challenges. And keeping this in mind, we need to work ahead. So before the end, so the team from the NDRI, the people, we have these Dr. M.S. Chavan, under his guidance we are working. Dr. Sachinand Day, his laboratory is working on the genomic aspects on the designing and the screening of the animal, or there is any mutations. He is taking care of that. My colleague, Dr. M.K. Singh, we are working together on the assisted reproduction, particularly cloning, production of cell lines or animals. We take care. And Devika, she's working as a research fellow under this project, and she's taking care of all these plasmid preparations and the screening of the animal or amplification. Our team from my previous team from the CRB, Dr. Yadav, Dr. R.K. Sharma, Dr. Dharmendra Kumar, and Dr. Niti Punetha, both these people they are working on this buffalo cloning, and they also been taking care of, because this project is joined between the CRB and the NDRI. And what the research have been explained today, these 3 girls, they have been engaged in this activity. Dr. Seema, she is a research fellow at CRB. She worked on the myostatin optimization for the single cell or transfection or the generation. And the Sonu has been worked on this preparation of the plasmid or the screening or amplification, because these are all the plasmids. So they have been designing, guiding, and the cloning and expression. She did it. And this Kamlesh, she is in taking care of the CD18 project. So she has been working on the optimization of the CD18 gene knock-in in the buffalo cells. Thank you very much.
Thank you, Naresh, for sharing. Thank you, Naresh, for sharing your valuable information in buffalo cloning and genome editing.